Well, welcome back, everyone, and thank you for joining our first panel discussion. First of all, I would like to really thank as well Dr. Feigenbaum for a remarkable and inspirational story that he shared with us. That certainly inspired me and I think raised a number of the issues that we'll discuss in the remainder of today's sessions, including the two panel discussions. In this first panel, we'll explore rare cancer registries and their utility in clinical decision-making. I'm David Reese, the head of R&D at Amgen. I'll be the moderator of the panel, and we're delighted to have join us today Drs. David Miller, Gregory Reaman, and Ting Li, who will offer perspectives from academia, the FDA, and industry, respectively. Before we dive into the discussion, I'd like to start with having each panel member provide a brief introduction, and perhaps, David, we can start with you.
Good afternoon. I'm David Miller. I'm a medical oncologist and dermatologist at Massachusetts General Hospital. My clinical and research focus is in cutaneous oncology. I am the director of the Center for Merkel Cell Carcinoma at MGH and co-director of our multidisciplinary non-melanoma skin cancer clinics. I've been working with PDS for the last 2 years on developing a registry for Merkel cell carcinoma. I co-chair that task force with Mike Wong, who you'll meet in panel 2, and it's a real pleasure to be participating in this along with Dave, Ting, and Greg.
Great, thank you. Greg? Yeah, you appear to be muted, Greg. So why don't you go ahead and unmute? There you go.
Hi, I'm Greg Reaman, pediatric oncologist. Sorry for the technical snafu there. Currently associate director for pediatric oncology at the FDA's Oncology Center of Excellence. I have a long history before coming to the FDA in clinical investigation in childhood cancer with a special interest in new therapies for children with acute leukemia and also in drug development for childhood cancer. So it's great to be here. Had wonderful experience over the years working with Project DataSphere. I'm happy to be part of this.
Great, thank you. And Ting?
Hi, my name is Ting Li, by training Hemong guy. Currently working for EMD Serono. I am the indication lead for Merkel cell carcinoma and GTN. Both are well qualified rare disease, I believe. So glad to be
really
here.
Looking forward, I think, to a rich discussion. As we mentioned, Dr. Feigenbaum introduced and touched on a number of concepts, but I thought perhaps we could start by going back to basics and simply start with a simple question. What is a rare cancer registry? Is it simply a database? Is it more than that? Are there flavors? To give all of our participants, a sense of the span of what we mean by the term registry. So David, perhaps we can start with you and get your perspectives on this question.
Sure, happy to. And I will start with basics as well. I've always liked the adage that if someone has said it better, then use their quote. So I'll quote E.M. Brooks, 1974, the current and future use of registries and health information services to sort of lay the foundation. He defined registries as a file of documents containing uniform information about individual persons, collected in a systematic and comprehensive way in order to serve a predetermined purpose. So a cancer registry, as I see, is an information system designed for the collection, storage, and management of data on persons with cancer. And I think about it in a couple of different flavors, to use that term. You can have population-based registries, hospital-based registries, and then special cancer registries, which I've been a part of, where the goal is to collect and maintain data on a particular type of cancer. And to also, I think, address your last question about the difference of a registry and a data set, I would say, and interpretations, of course, vary, To me, the difference is a registry is a special type of data set that has a longitudinal component that is critical for the study of the natural history of an illness. So it is that ongoing nature, the continued curation over time that kind of gives a registry its distinction from just a plain data set that may be static over time.
Great, thank you. No, that's fantastic. Ting, maybe from the industry perspective, your view of what is a registry. I know you've had experience in this regard.
Yep. Thank you for the opportunity. Quite frankly, for the industry, used to be, or traditionally, it provides safety signals for the drug, especially as Dr. Fajgenbaum actually referred to the phase 4 studies, and used to be used for monitoring AE events and to validate the efficacy and the safety information, mostly focusing on safety. However, in the current practice, especially in oncology, that's how it can be used, how it can be informative data set for everybody, for prescribers, for industry, and for scientists in the lab. It's actually a treasure trove. That's what I can see. David already touched upon the longitudinal aspect of it, and that actually will be very informative in terms of a sequencing of the treatment, concurrent treatment versus sequencing. What will be providing the best efficacy? What will be providing the best safety profile? That's actually an excellent question to ask in the registry. And also I want to stress the point of comprehensive and systematic. As the keynote speaker already touched upon, and also David touched upon, sometimes those kind of information can be really ignite a fire by a sparkle of a biosample study and a cytokine study. So this is actually could be a very informative.
Thank you so much. And Gregory, from a regulatory perspective, any thoughts on sort of a basic definition of a registry?
Basic, I think David and Ting actually both contributed to a basic definition, if you will. I think one of the really critical issues in defining the basic necessities of a registry are sort of defining a purpose for the registry, the longitudinal nature of the registry, and most importantly, when aggregating information and particularly patient experience, doing it in such a way that that information is going to be operable, interoperable, and publicly available and utilizable. Utilizable by patients and families, utilizable by medical practitioners and caretakers, and ultimately, I think utilizable as we think about what is clinical benefit, and looking at the heterogeneity which spans rare diseases, except maybe those rare diseases which are due to a single gene defect. But given the variability in symptoms and clinical manifestations, I think having a registry and looking at potential interventions or just looking at natural history without interventions can help define what patients might experience and what patients might consider benefit. And that consideration really needs to drive drug developers and ultimately regulators.
Great, thank you. Well, you brought up sort of purposes and goals of a registry, which really, leads naturally to the question of what is the intent of a registry. Or, of course, there can be perhaps many intents, from charting the natural history of the disease, as we heard from Dr. Feigenbaum as a first step, all the way through potential roles in regulatory outcomes. It'd be good to hear from our panelists again about their views on intents, and how do you think about structuring a registry when you're thinking about the end in mind. So David, you've got some experience here. I think it would be valuable for you to share that with our audience because you've worked through a number of these problems yourself.
Yeah, thank you. It's a great question. And thinking back to 2 years ago when our task force first charted out our course, a key question, like the key question, was what are the objectives and applications? And over time that has evolved, not surprisingly. Initially, the idea was we had a rare disease in front of us that we all treated, but there was little data known about it. So understanding the basic characteristics of people who developed Merkel cell, and then understanding how the disease, how it progresses, what I would classify as understanding the natural history of the disease was initially, I think, the primary focus. And then, as we'll probably talk about later, that's evolved a little bit to also incorporate information potentially on best practices. I think Ting touched upon some of the best practice questions that we have. How do we sequence the available therapies that we now have through innovation at the sponsorship level and innovation at the regulatory level, to potentially getting information that could affect regulatory decision making, potentially affecting label modification through our data. And having those objectives thought out ahead of time is critical when you are developing your instruments in data fields in order to collect those important data elements to achieve either natural history understanding, best practice understanding, or potentially to influence regulatory decision making.
Great. No, thanks. And I want to return to that topic of best practices in just a minute. But Ting, interested to get your take here on intent, based on your extensive experience as well.
Sure. What I would think is actually natural course of a disease will change with different treatment options. A good example is actually Merkel cell carcinoma as well. So with the advent of avelumab and a checkpoint inhibitor in that particular rare disease, the natural course of the disease has been changed dramatically. So what we raised the bar of the survival of the Merkel cell carcinoma, let's put that way, and how high the bar is, and what will be the follow-up treatment should be improved upon, we don't know. So that's why even if we just a simple intent of capture the natural history of the disease is actually very informative for follow-on improvement and drug discovery as well. And let alone the combination, as I've already mentioned in an earlier conversation. So this is actually could be very constructive going forward. And for rare disease, actually the real world evidence could serve as a comparator arm. Quite frankly, because it will be very difficult to actually have a controlled study design for future regulatory approval. So this is just the couple points I want
David
to cover.
brought up a potential regulatory role of registries, and so we're interested in your perspective from the FDA. What do you view the potential role? Are there different roles? And I think this is a question that our audience will be intensely interested in. It's obviously a wide-ranging question, but we'd like to get your point of view.
Sure. Well, I think this topic will probably be also covered in the next panel. But I think as a start, I think there are multiple different opportunities and potential contributions that registries can make to the regulatory setting. Defining potential endpoints that might be used in subsequent clinical trials based on outcomes of patients and based on the natural history of the disease in patients. So just determining, what is a patient's perspective on what would be called a clinical benefit or classified as a clinical benefit. I think from natural history would certainly give us an opportunity to look at endpoint development. One other thing I would go back to Dr. Feigenbaum's presentation about the linkage of registries with clinical data to biobanks. I think here again, there's a very rich opportunity to look at material that is available and usable again, and usable by scientists in multiple sites to actually look at correlations of different characteristics and natural history and potential biomarker development. As he very eloquently portrayed, not all patients with multisystem Castleman's respond to IL-6 inhibition. So looking for specific biomarkers that may predict a response to a new therapy, I think is an important outcome from
Great, thank you. And again, a lot of rich material, some of which our second panel will also explore. So we've talked a lot in theory so far, and perhaps we can move to actually some practical examples. If our panelists can give us examples of registries that they think are high quality and have utility. And then from there, perhaps we'll move on to what we can extract from best practices from those examples. So, let me start again with David here in terms of, when you survey the landscape from 1974 on, what sort of registries jump out at you, whether they're within oncology or outside of oncology for that matter.
Perfect. So, there are a plethora of examples where registry data has influenced my practice, and that's in part because I take care of very rare diseases, and Merck, so as has been said, is a great example of that. There are very few clinical research studies in general, period, in Merck, but some of the ones that have influenced our practice come from, for example, SEER, the Surveillance and Epidemiology and Results Registry, which has been conducted through the National Cancer Institute. The National Cancer Database, or NCDB, is another rich source where numerous papers have been published using that data to influence our practice. And just for example, the current staging system that we use in Merck Cell, the AJCC 8th edition, which informs how we deliver prognostic information to our patients and how we ultimately adjudicate treatment decisions, came from a data set from the National Cancer Database. 9,000 patients with Merck cell were used to construct the most recent staging system. So that's a good example of very practical applications. I have two more that I can briefly cite as well. So in Merck cell cancer, there's a standard of care now to treat patients after you've had surgical excision with adjuvant radiotherapy. For most cases, there are some nuances to this, but that current standard of care of adjuvant radiotherapy in patients with MCC in part was supported from a 2007 publication of 1,100 patients from SEER, and then a 2019 publication of another 4,800 patients from the NCDB. My current practice of not using radiation for low-risk Merkel cell carcinoma patients came from a data set from the University of Washington, their single institution data set influenced, really changed my practice. And then the final one of using baseline imaging in patients recently came from a publication that we actually just reviewed yesterday in our multidisciplinary journal club that again came from a single institution registry from the University of Washington, 500 patients, where they said, you know what, unlike melanoma, which is where we've been getting a lot of our practices, sort of inferring practice management of patients from melanoma into Merkel cell. Now, in contrast to melanoma, which typically, unless patients have regional and clinically regional involvement, we don't do baseline imaging. In Merkel cell, there's been heterogeneity in practices. And a recent publication that hasn't even been in print yet has sort of affirmed my practice of actually using baseline imaging in all patients with Merkel cell, because 1 in 3 patients that walk in the door with Merkel cell have either occult regional or occult distant disease. And that came from a single institution registry. So for rare diseases, registry data is pivotal in how we practice.
Great, thank you. Well, maybe we can go to Gregory next on this topic. You know, from your vantage point, you've seen a large number of registries. I wonder if you could share with us what you think some of the highlights are in terms of best practices.
I think as far as best practices, and one registry that particularly comes to mind, as a pediatric oncologist, all pediatric cancer is rare, but there are the rarest of the rare pediatric cancers for which clinical trials haven't existed, don't yet exist, but there are clearly opportunities. And one such registry is in a rare pediatric tumor, pleuropulmonary blastoma, which has, I think now, in over 20 years, basically accumulated a large natural history data on a sizable number of patients. There are only a handful of such patients diagnosed yearly, but it has really resulted in the development of the standard of care, if you will, from the standpoint of diagnosis, both clinical diagnosis as well as pathology, and now the banking of specimens to look for specific biomarkers that I think will actually result in the development of targeted therapies for this disease. So I think having a recognition that, and I think critical to best practice, is really a willingness to collaborate, participate, and understand the strength of coordination, cooperation, and collaboration. And in rare diseases, obviously that's critical. In childhood cancer, that's what's really driven the field over the last 5 or 6 decades. I think a realization and acceptance of that is
great. Thanks. Ting, anything you would like to add in terms of best practices based on your experience?
Sure. Before I joined the industry, I actually was working for Janssen on a CLL program, so I was involved in some CLL registry. What actually has been noticeable in terms of a registry in CLL is actually the contribution from the community practitioners. So we talked a lot about biobanking, we talked a lot about research, but the simple community, not cytogenetic, not biomarker-driven kind of clinical practice need to be captured as well, because that represents some probably underserved patient population as well as part of the real world. This is actually just the one point I want to just make sure.
Great, thanks. And maybe we'll start with you, Ting, on this next topic as well. We've got a number of questions coming in, and one of them I think we want to address because it will be of great interest is, what are the challenges in incorporating data-rich data-rich elements in the registries? I'm thinking of omics data or immunophenotyping or imaging, which of course requires large amounts of storage and ways to handle the data and to ensure data quality. So Ting, perhaps you can give us your perspective, again, having established some of these registries and trying to encompass molecular and imaging data as well as basic patient data.
Sure. One thing I can think of regarding imaging is actually standardization. Several co-op groups actually had went great lengths to actually standardize all the imaging technique. That's actually quite crucial for the uniform readout for the results and assess the efficacy. So kudos to all the radiologists who is actually doing that. That's actually crucial for us as well. So that's one thing. Another part is actually personalized medicine. Personalized medicine really is actually individually tailored treatment. It's very difficult to harmonize, and hopefully the big data, we are talking about the PDS here. So the algorithm behind the big data readout could help for everybody to sift out some clue in the treatment. It's actually very heterogeneous in terms of cancer, in terms of response to drug treatment, and hopefully some clever algorithm can find us.
David, how are you thinking about data beyond basic natural history data, omics, imaging, other types of intensive data?
Yeah, so I think there is what I've been thinking about a lot lately is there's two opposing vectors when you're doing database or registry design. And there's the vector of data-rich data collection, and then the vector of feasibility. And a lot of times those are in opposite directions. I think all of us want to capture as much data as possible so we can move the field forward. And our task force has had lots of conversations and, curation, rounds of curation in order to enhance the feasibility of this. So, we've taken the approach where we've had to early on prioritize which large data that we are able to capture first. So we've taken the approach of a multi-phased sort of data collection and release process. So initially, we thought the highest yield data the data element that found the right mix of feasibility and clinical utility, sort of capturing genomic data. So what we've done is develop an instrument in order to capture commercial and institutional NGS data. And then this is data that we can abstract from either a health record or from a report from a commercial vendor. Most of the time it's unstructured data, sort of turn that unstructured data into structured data and then import it into our data collection tool. And that requires, some data science skills, it requires some resources, and also requires some skill. So we're hoping that our experience through capturing genomic data is going to inform our ability to capture the next phase of large-scale data, which, imaging is something that we're very interested in capturing serial imaging on patients in order to be able to perhaps inform, regulatory decisions based on imaging. Now, at this point, our imaging capture system is based on data entry of discrete fields. And for radiology reports, that requires pretty intense resource in order to go through radiology reports and sort of do manual abstraction of those reports. With time and data science solutions, we hope to be able to innovate and be able to come up with text mining solutions in order to facilitate that. But at this point, we're still doing some manual abstraction. So in order to continue to make progress, we've sort of decided this would be a multi-phased approach where we prioritize capture of genomic information. And then once we do that, we'll sort of move towards the next phase, which would be imaging data.
Great, thanks. Well, Gregory, assuming we surmount all of these various challenges, is the FDA ready to, ingest large amounts of registry data? And it'd be very interesting to get your perspective on, how, we as a field can best utilize these data, but also, just from a regulatory perspective, how you'll actually handle it.
Good question, for which I don't have all of the answers. Is the FDA ready? I think the FDA has to be ready to at least work with industry and work with investigators and patient groups because we recognize the importance of these data and their potential contribution to future regulatory decision making. As some of the challenges have already been pointed out with respect to infrastructure and resource capabilities, the agency obviously has similar constraints. But I think working together as a field, we recognize that in order to get drugs to the patients who need them the fastest, we do have to look at new ways of demonstrating clinical benefit, new ways of determining short-term, long-term toxicities, and the old ways that we have done that in the setting of rare cancers really needs new think. And I think moving forward and working together, utilizing registry data is something that will become part of what we...
Great. No, thank you. Well, and another question that's come in, which I think will also be of great interest to our audience, is the notion of open access. Project DataSphere, of course, pursues an open access model, but not all databases are open access, nor are all registries. Perhaps, David, we can start with you. The specific question is, how do we overcome institutional or organizational barriers, to submission to open access registries? It'd be very interesting to get your perspective from academia. Then I'll turn to Ting, who will give us perspective from industry, where, of course, other considerations also apply.
So it's a critical question, and one that our task force has been very thoughtful about. And there's no perfect solution to overcome some of these barriers. One methodology that we are moving forward with is sort of understanding that we all have various and different incentives. We've developed a multi-tiered data release platform that we are moving forward with. It's basically 3 tiers. So the first tier is a closed data collection and data access phase. So with our capture system, each institution that's led by a principal investigator is sort of the governor of that data set. And that individual institution has the ability to collect their own institution's data and then publish it as a single institution data set if they wanted to. Now, we realize that there are limitations to single institution data sets, and all the investigators in the task force realize that there are limitations to single institution data sets. So we know that most people are going to be motivated and incentivized to move on to the second phase, which is more of a closed access to the individuals who have aggregated that data. So for example, in the second phase, data that is captured by each institution is migrated over to the PDS platform. There's a period of time where that data is available to the institutions that have contributed that data. So there's a governing committee that would adjudicate the hypotheses that would be tested on that aggregated data. And then after that phase, we would open it to registered users on PDS. And we think that multi-step model provides the incentives that are necessary for investigators to participate. All of the investigators at each site have lots of different diverse teams that have different deliverables that they want, and having the ability to publish a single institution data set so that a graduate student can get a PhD dissertation, is an important option for people to be able to have, for example. And then having a data set that is aggregated gives incentives to the investigators. But then, of course, ultimately, what we want to be able to do through PDS is provide that information to a wider audience so that they can ask and test hypotheses that, we haven't thought of, or we won't think of, in order to move our field forward. So that's one of the methods that we're using in order to try to align incentives within the realities of academic medicine.
Yeah, no, that's great. And that's, it's a very interesting thought around a period of restricted access for those who have contributed data and sweat equity, then followed by open access, a way to sort of compromise and provide appropriate incentives. Well, Ting, from your perspective, what are some of the barriers in industry? Of course, there may be proprietary considerations and other issues. How do you resolve those sorts of questions within an organization?
Yep. Thank you for the question. This is actually a challenge for every one of us. I cannot commit to say that we will disclose proprietary information that will be still protected. But frankly, by principle, it's actually to improve the health, improve the clinical outcome for the patient. So by that particular principle, we are committed to as much open as we could, especially on the safety signal side, on the efficacy outcome side. For sure, you have our commitment. Our senior management team is also team member, are also on this particular call. And I think he will agree to that as well, that it's a serious commitment from a industry partner that we will be open for the community, not only for the treaters, to the researchers, but also to the patient.
Really valuable perspective. We've got another question here that I think will be of great interest, which are, what are key considerations when you're using registries like SEER, NCDB to derive insights for broad populations? In other words, can we rely on registries for generalizability? What are our thoughts about that? Are there pitfalls? Are there ways that we can protect against false positive findings? Obviously, the larger the data set and the more clever the statistician, the more likely one will find associations. The question is whether they are true or not. And that's what interests all of us. And so I'm interested in this, sort of notion of how do we rely on registries for generalizability and the soundness of our conclusions. David, maybe we'll start with you here, and then we'll go to Greg, from an FDA perspective.
Yeah, I think those are great considerations, great questions. And, it's a multifaceted answer, of course. There are limitations to every single data set, and appreciating those limitations, whether they're missing data, biases, confounding, are critical. So having those registries be very transparent about how they have handled missing data, for example, or how they've handled bias is critical. And I think as a practitioner, one of the things, so at the core of clinical research is trying to minimize medical decision-making under uncertainty, right? On a quotidian basis, I am, and all clinicians are carrying out medical decision-making under uncertainty. And therefore, in order to minimize that, we need reliable inferences from data sets. And I think what at the core is, there is no single reliable data set, right? We need to pool multiple data sets and be able to synthesize and integrate inferences from each data set in order to ultimately, take care of our patients, in order to ultimately make medical decisions. So there is a real need to have numerous registries in order to have, say, validation of the findings of one registry. So if we can support some of the findings in SEER or NCDB with our registry, then that provides confidence in those inferences. And as you said, as we sort of grow our data sets, we get closer and closer to big N. We have more reliability on those inferences. So I think, to summarize, being very transparent about missing data, biases, and then also understanding that we need multiple various sources of those data in order to have reliable inferences to make medical decisions.
Oh, great, thanks. Gregory, from your perspective, what do you look for in terms of comfort with generalizability or in assessing the soundness of findings that may emanate from a registry?
Well, I think from the perspective of generalizability, and particularly since we are discussing rare cancer registries, I think a registry that draws from more than a single institution is clearly a superior registry, only because of or not only, but in large part because of the concern of unintentional bias that may exist, and especially when data elements may be objectively determined data coupled with subjective assessments. I think unless the registry is very, very well structured and that structure is made transparently known to potential users, then I think having a registry that draws from multiple centers, multiple treatment sites, patients from different parts of or geographically dispersed and represented, I think would give the agency more comfort.
Great, thank you. Well, another question that's come in relates to patient-reported outcomes, and I think we'll have time to address this final question. And it's appropriate that we return the focus to the patient, of course, because we want to improve patient care and patient outcomes, when we are constructing and utilizing registries. And one of the key things for patients, of course, are what we call patient-reported outcomes, that very broad category of data and assessments that include quality of life and many other measures. So the question is, how do we think about patient-reported outcomes as we're generating registries, and how do we use PROs to really inform us about the natural history of the disease, but how patients are actually really deriving benefit, for example, from potential interventions. So, David, why don't we start with you here, then we'll turn to Ting, and we'll wrap things up with Gregory. David?
Perfect. So it's a great question. I think patient-reported outcomes are a key metric for healthcare delivery. I think that the question highlights a couple of things. One is that, when you're constructing a registry development team, it's really important to have a diverse group of experts on your panel. And you need to have domain expertise, of course. In the case of Merkel cell, you need to have dermatologic expertise, pathologic expertise, medical surgery, radiation oncology. But beyond that, you need to have expertise in data sciences, biostatisticians, epidemiology. And I think patient-reported outcomes is a good example of a niche that requires a lot of expertise. And, patient-reported outcomes are facilitated now due to some of the technological advancements in data capture systems. So the data capture system that we use, REDCap, has the ability to have surveys that go out to respondents or patients, and then they're able to fill out that data, and then you can insert that back in a structured form into your data set. So that is something that we haven't integrated yet. It is a plan for 2021 is to sort of figure out ways to optimize PRO assessments into our registry. But at this time, I haven't had the time to really dive into it. So a real nuanced discussion of PROs is a little bit beyond my expertise at this point. So I'd be happy to get some insight from the other panelists.
That's great. Thank you. Ting?
PRO, patient-reported outcome, and the quality of life assessment is inherently part of the registry if industry is designing it for two reasons. One is actually just as our chair, David Reese, just mentioned, it's actually all about improving the quality of care. Another aspect is the payer. How are we going to justify certain cost of the healthcare to the payer system? That's another endpoint. And increasingly, the quality of life becomes a regulatory endpoint as well. I believe Greg can touch upon this. At least in Europe, this is becoming more and more
Thank you.
important.
Gregory, why don't you wrap things up from an FDA perspective on PROs?
Well, I think PROs are clearly something that are of great interest to the agency, and recognizing, appreciating, understanding, utilizing, hearing the patient's voice in regulatory decision making is really critically important. I think there are difficulties with validation of specific instruments that might be used. And I think another challenge is sort of global quality of life measures, which may be impacted by much more than the disease itself or the therapeutic intervention. And how does one really separate one from the other? So I think in designing registries and considering PROs, as I said, I think in the beginning, part of what I think a registry can really contribute to understanding natural history, or what the natural history can really help investigators and sponsors understand, are what is the potential benefit that patients are seeking with the illness, the rare disease, or the rare cancer that they might have, and then utilize that information to design, develop outcome assessments that can be standardized, validated, and used in a setting where the data collection and management, or the integrity of that is not going to be questioned. And I think going forward, that's what we would...
Great, thank you. Well, I'd like to thank all of our panelists for an engaging and very informative discussion. A number of these points will be taken up by our second panel. We heard about basic definitions of registries all the way through considerations of methodologies, and then the intents and uses of registries. Next, we're going to take a 5-minute break. And then after that, panel 2 will resume our discussion of rare cancer registries, specifically in the context of drug development. So we'll see all of you again in 5 minutes. Thank you.